The cell wall is a type of extracellular matrix which plant cells secrete around themselves. Most cell walls are composed primarily of cellulose, a polymer consisting of thousands of glucose monomers, although more rigid, woody cells contain rigid networks of lignin deposited in the extracellular matrix. In this diagram, the blue bars represent cellulose microfibrils which provide the tensile strength of the cell wall. The green strands represent cross-linking glycans which hold the cell wall together and keep it from falling apart. The pink strands represent a molecule called pectin, which merely takes up space between the microfibrils and provides stability to the cell wall. Cell walls are necessary in plants because plant cells lack the intermediate filaments which provide stability to animal cells. Like all extracellular matrices, the cell wall derives its strength from long fibers oriented along the lines of stress. Without the cell wall, the plant cell would be likely to burst even at the slightest shift in osmotic pressure.
The extracellular matrix, also known as the ECM, is made up primarily of glycoproteins, which are proteins covalently bonded to carbohydrates. It is made of collagen, which are strong fibers outside the cell. In this diagram, you notice that the collagens are the long lavender tubes. It is also embedded in a network woven from proteoglycans. In this diagram, you notice that at the upper left-hand corner, it is the strings attached to the polysaccharide molecule. Some of these cells that are attached to the ECM by glycoproteins are called fibronectin. Fibronectin and other proteins bind to the integrins, which are at the bottom of this diagram in the purple, and that are built into the plasma membrane. It helps aid in transmitting charges between the outside of the cell and the inside. It regulates cell behavior, and the extracellular matrix of a particular tissue can coordinate behavior of all cells in that tissue. It is located outside the cell, hence extracellular, and communicates with the cell through integrins binded to fibronectin. It works with microfilaments in the cytoskeleton to help the cell react to external stimuli. Without it, we would not be able to communicate information between the cell environment and the internal cell organelles. And an interesting fact about it is that it influences the activity of genes in the nucleus.
A cell junction is a type of structure that exists within the tissue of some multicellular organisms. Cell junctions consist of protein complexes and provide contact between neighboring cells or between a cell and the extracellular matrix. There are 5 main types of cell junctions. Tight junctions, which are exclusive to epithelial tissues, seal neighboring cells together in an epithelia to prevent molecules from leaking out between them. Adherence junctions join an actin bundle in one cell to a similar bundle in a neighboring cell. Desmosome junctions attach the intermediate filaments of one cell to those in a neighbor. Gap junctions allow for the exchange of water and small ions between 2 neighboring cells. They consist of connexins, which are protein complexes which stretch between the 2 plasma membranes. This creates an electrical and metabolic coupling between the cells. Gap junctions between heart muscle cells provide the electrical coupling that allows electrical waves of excitation to spread through the tissue, triggering coordinated contraction of the heart muscle. In conclusion, much of the intracellular communication that takes place would be impossible in the absence of cell junctions. Rapid muscle contractions, and by extension human locomotion and heartbeat, would be made impossible. Therefore, cell junctions were a crucial evolutionary step in the development of multicellular organisms.